hear next from Yael Rosenberg-Hasson, who is our immunoassay manager in the HIMC. She's done a lot of work with Luminex and also MSD assays, which you'll hear about, and is going to basically tell you today about some of the things we've learned on these systems, how they compare, and how we use them. Thanks. Yes. So this goes here.
So hello everyone, and I'm Yael, and I'll be describing two of the multiplex immunoassays that we run in the lab, namely the Luminex assay and the MSD assay. And just to mention, we would like to measure cytokines. Cytokines play a key role in our immune response. And therefore, since they are present in very low amounts in steady-state levels in our system, we need very sensitive methods to detect them. And so these two methods offer you a broad range, a broad dynamic range to detect these cytokines and chemokines. So I'll start with the Luminex. And the Luminex basically is a flow cytometer. It's a dedicated flow cytometer where the ELISA, the multiplex ELISA, is built on a bead. So first of all, these beads are polystyrene beads. They're dyed to create spectral addresses. And those spectral addresses determine if it's going to be an IL-2 bead to detect IL-2, TNF-alpha bead, etc. We currently measure up to 55 different types of cytokines and chemokines, actually 51, 4 of them being control beads. We also measure, that's on polystyrene beads, we also measure on 39 targets on a magnetic type of assay, similar. And what happens is that on that little bead you build a very nice ELISA on that bead that is detecting a specific cytokine. So your sample is captured here, then you add the detector, then you add the fluorescence tag. Now, when it flows through the cytometer, the red laser detects what type of bead it is, and the green laser detects the amount that is bound to that bead. And that's basically the assay. So the MSD is another multiplex type of assay where the ELISA is built in the bottom of a 96 well plate, where the plate is a pre-patterned array. So you have up to 10 different cytokines and chemokines that can be captured at the bottom of the well, and then your assay is built in proximity to that captured antibody. Later amplified, this is an electrochemiluminescence assay, which allows you later to amplify the signal and to emit the light that is detected by the CCD camera. It's called the sector imager. We call the platform MSD for the company's name, but it's actually, the instrument is called a sector imager. So here you can see what happens in one well. You see that here the pattern is of a flower of 6 different cytokines that are detected. The nice thing about this system is that it's not a flow cytometer, so the samples do not clog the probe, and the plate is read in pretty fast, and the assay is a little faster. The limit is obvious. The limitations are the fact that you can analyze only up to 10 in a plate, or 1. But it's a very nice system that it basically should be replacing any ELISA that you've been doing, because it's very sensitive and offers you a very broad range. The one drawback on the system is that it reads in sections. And so if, for instance, you have 6 cytokines and you have one cytokine that has a very high level, it's going to compromise the signal of the others. So if you find that, you may need to modify your kit. So this is summarizing a few of the parameters. Basically, the nice thing about the multiplex assays is the fact that you can take a 25 microliter sample, liquidified sample, and put that in your array, whether Luminex or MSD, and get an output of up to 55 cytokines in the case of the Luminex, up to 10 in the case of the MSD. Again, we report median fluorescence intensity and picogram per mil. I'll elaborate shortly. Again, here we report the light intensity and the result in picogram per mil. So what type of fluids can you analyze on these systems? So basically, this is not a complete list of the samples that we've analyzed, but basically any liquidified sample could be analyzed on these systems. And the most common ones that we've analyzed are the serum and plasma, cell culture supinatants from all sorts of cells, and tissue homogenates, basically mouse samples that I have not mentioned, but we do offer a 26-plex mouse array. Yes, and actually, plasma and serum are the most abundant and common ones for the human analysis. So what type of controls do we offer you? And I'm elaborating on that part a little more in the demo today. I will go into more detail. But basically, for the Luminex, here what we do is we usually run replicates of 2 per sample. We look for CVs that are lower than 10%. In most cases, they are lower than 10%. Only very few would have a CV, a coefficient of variation, that is higher than 10% or higher than 20%. We use, as I was sharing, we use 4 additional control beads that help us QC the steps of the assay, making sure that we entered what we should have entered in each step of the assay. The other thing that we've come to add into our assays is a control sample. And we prefer a serum control sample because that's a biological sample that can offer us comparison and to follow our reproducibility and performance over time. So this is just the presentation of the 51-plex, of which the ones in red are the ones common to the MSD and the Luminex, and I'm going to elaborate more on them in the next few slides. Again, just to mention, looking at our control sample, we're going to concentrate on our control sample in the next few slides. Here, what I need to mention to you is how do we report the data? So the data is reported as the raw signal. In case of MSD, it's light intensity, and in case of Luminex, it's the median fluorescence intensity here on the Y side, on the Y-axis. Now, how do we convert that into a picogram per mil concentration? So the way we do that, we include standards. That's a known amount of those clean cytokines prepared fresh in buffer and aliquoted. Then they are serially diluted to create this standard graph. That's a 7-point standard graph for each of the technologies. It's not the same standard in each of them. It's not common. Each platform has its own. And what you can see, this allows you to convert the raw signal to a relevant concentration in picograms per mil. And I'm going to spend some time in the next few slides looking at our control sample in the light of what I've shown you here. So what do we recommend you to use downstream from the data that you receive? And so if we look at I'm first going to describe the Luminex, and if I take that control sample, it's a serum control sample that was run here. I'm showing you only 3 repeats over a period of 3 weeks. And what you can see here, that if we look at the reproducibility of the data at the level of the MFI, you can see that it's pretty much reproducible. I'm only showing you some of the cytokines, and this is just these 9 cytokines that are common. Then if we look at what happens when we convert the signal that we got here to using our standard curves to concentration, what we see is that we get much more variability in terms of what it's detecting, what it's presenting as the result in picogram per mil. So this is true. It turns out to be true not only for the Luminex, but also for the MSD. Again, that same control sample, actually from that same plate, we ran side by side both technologies, yielded the same picture, so where you can see that the reproducibility at the level of the intensity, the raw signal for MSD, is much better than the concentration picogram per mil. So what do we think is happening here? So just to give you our view of what the standards are doing. They're intended to help you normalize the data and present the data in picograms per mil. But actually, when you think about it, they're creating more variability for several reasons rather than normalizing the data. One way to look at that is to look here at the standard graph for IL-6, and you may be able to appreciate that fact that a small change in MFI would lead to doubling or tripling of the value given by the concentration, picogram per mil. So that's one source of variability. There are others, right? We're doing everything we can to be as reproducible as we possibly can, and that includes using standards that are prepared as a stock, frozen in -80 in aliquots, so to rule out that variability. And then we thaw a fresh aliquot every assay and produce that standard curve. Still, the dilution day to day, even though it's the same even person, it's not always going to be exactly the same. So there's some variability that we have to live with. But generally what we see is that our raw signal is much more reproducible than our picogram per mil. Therefore, I hope I've convinced you to use the raw signal rather than the picogram per mil. So the next issue that we were testing in our system was how does the matrix of the sample affect the detection of our cytokines. Remember that what we're doing is we're taking standards. That's another measure to why we're not sure about the standards. We're taking clean cytokines and chemokines, suspending them in a buffer, and assuming that that will mimic what's going on in serum, plasma, homogenates, etc. And we know that that's not the case, and this is a spike-in experiment where you can see that. So here, again, looking only at a subset of the cytokines, in the buffer, this is the level of detection per cytokine. If we resuspend that same standard, that's standard number 6, in 4 different serum samples, what we see is a reduction, a very significant reduction in the detection of the signal. So this shows you that there's some compromise. The fact that you're detecting, the biological sample is a much more complex sample, therefore there's some quenching going on there. However, that's not the entire story. It could be inhibitors, etc. So what would happen if we would dilute the sample? Would that improve our odds? Is it concentration dependent, this inhibition? And indeed, we see that in some cases, for instance, for this subject, what I need to explain to you here, these are 6 different subjects, and we're running a neat 2-fold dilution, 4-fold dilution for each one of them, looking at IL-6, IL-8, and TNF-alpha. So there is a decrease in the detection that coincides with the dilution. However, for instance, in certain cases, for instance, this Superman here, we call him Superman for several reasons. He has high levels of cytokines. And you actually see an increase, so that there's an inhibitor that upon dilution you get rid of, and it's easier to detect this cytokine. So in Luminex, we think that dilution is definitely a good option to prevent going into reaching those problems of inhibitory factors. What happens in MSD? So the same spiking experiment where you take the S6 standard and place it in serum or plasma, because here you have one in serum, one in plasma, you see that for the MSD platform, that's not the case. There's no inhibition of signal. What we think is happening here is, well, some of the explanations include antibodies, and we think that's a combination of several of them. But basically, one of the differences between MSD and Luminex is the fact that in the MSD, the first antibody is captured on the bottom of the well. Therefore, there's less interaction, much less interaction that's happening further downstream. It's not the entire story, but it's part of it. So in summary, what I think I've shown you is that even though we can detect cytokines in picogram levels, in the picogram level, which is required for monitoring the immune responses that we're looking at, we recommend to use the result, the raw signal that is either the intensity or the median fluorescence intensity, just because that is much more reproducible. And we see an inhibitory signal by serum and plasma, for that matter, that is somewhat reduced upon dilution, and it's seen in Luminex, not in MSD. Another very important point that I would want to make here, and I did not mention one important one that I'll mention in a second. So I want to remind everyone that chooses to run their samples. Upon your collection of samples, adhere to your protocol, aliquot up front, because then you save freeze-thaw experiments. I haven't mentioned that, but freeze-thaw is a no-no. So we would like to be at the point where we run samples, we freeze-thaw samples no more than 3 times at most. Preferably only once. So upon collection, you would want to aliquot your samples, freeze in aliquots, and use different aliquots for different assays to be as close as possible to perfect in terms of the monitoring of the level of the cytokines. So where's the future? The future is actually, again, going to the normalizing of the data and the data analysis. That's always a challenge. And you want to try and analyze the data as best you can, looking at multiplex data, still preserving the rainbow of the variability of that data. And I want to thank Iris and Cindy, who've been doing most of that work, and all the HIMC members and everyone listed on the slide here. Thank you very much, and I can take some questions. One thing I did not mention is, more data, detailed data analysis. I'll be demoing in the afternoon, showing you how we produce our reports, what QC steps do we take, what do we look at, etc. And I think that could be very informative for those of you looking at those reports.
Yeah, maybe I missed this point. Is this set of cytokines the same in Luminex as well as in MSD, or do they
So while I showed you right in this slide here,
51 black.
mhm. So here, the ones in red are the ones common to the MSD and to the Luminex. So we measure them both here and here. However, that does not mean that they're using exact same antibodies. They're produced by different companies, and therefore the antibody pairs, et cetera, are completely different. So not necessarily the same antibody would be used for both cytokines. And that's another measure of variability, definitely.
What's the cost per assay? And if you are doing like a supernate from a cell culture, would you recommend doing your concentration dilution in like the basal media for that cell culture? Or?
That's a good question. So basically, the cost is an easy one. I think if you're on a fishing expedition looking for the 51-plex offers you a better chance of finding something. And again, per cost, the MSD hasn't got a cost per sample. It depends on the kit that you want to run. There's a cost of labor that goes in together with the cost of the kit, and that's something that we can offer you. It's cheaper than Luminex overall, but you get much more for a little more than double the price. The price for the Luminex is $105 per sample. That's for 2 replicates. That's the first question. The second question was about using your soups. So your soups is a challenge. If you're just looking at steady state levels, you would not want to dilute anything. However, if you're stimulating your cells, you definitely want to consider that. You could run a neat sample and then dilute, or run side by side a neat and diluted. The dilution depends really on the type of stimulation. Basically, if you're hitting those cells with PMA ionomycin, from our experience, sometimes you need more than 1 round of dilution. So it could be a 5-fold, 10-fold, and as needed, because the range of the cytokines can vary considerably. And so you won't be getting all of them at that range if you wanted to use the picogram per mil. Mm-hmm.
So we have a panel here of 51 things that we can measure with Luminex. What if I want to add something to the panel?
So the panel as is, we typically do not add on to this panel. We run, we have this custom panel, we offer that one. We cannot take out. It's a pre-mixed panel. You could possibly add in, however, you do not know to expect what the influence will be in terms of the background level, what it'll do to other cytokines, and how it would be detected in that background. So that's something that the companies work on quite a lot in order to get them all within an acceptable range. So that's not something we would recommend to do. You could purchase your own and ask for tech labor. That's another option, probably.
Does the company itself produce all of these?
Yes. So what they do, they offer, well, there are different companies. EMD Millipore by far is a larger company and offers a much broader number of kits and possibilities. They offer you a plexing of different types of kits. However, I think you do, for your first fishing expedition, I would recommend to use the ones available. So either the 39-plex or the 51-plex. Unless you have a very specific set of cytokines of interest, and then you would want to come share with us, discuss, and see what we can do to help you on that. Mm-hmm.
So since the serum have the inhibitor to some of these cytokines, so what do you recommend? I mean, if you look at just one without diluting, or we need to do multiple dilution to do this luminous.
So you're thinking, yeah.
Kind of expensive.
Mm-hmm. You're thinking about an ELISA. What we typically do, and that's part of the reason we recommend not to use a picogram per mil. you have a pretty broad dynamic range of detection there. So if you run just the neat or twofold or fourfold or fivefold dilution, you should be typically within the range of most of them. So you would not need to repeat. Yes, if you wanted to insist on a picogram per mil, you would probably need to repeat. And that would cost you a little more. But then maybe you would prefer to run a single ELISA on that one, right? If there's any one of interest that you really want to quantify to that level. We think that the data is best presented as a fold change. And in the demo, you'll see that there's some loss of data because the standard curve actually cuts off. I can go here. We have another minute. Yep. Sorry. So what you can see here in the upper dynamic range, there are two problems there, right? In the lower dynamic range, so in the lower dynamic range, we probably at the basic level of detection, we cannot go beyond that. If you dilute, you lose your signal. However, let's say the MFI was 20. On the lower end, you cannot dilute further. So you would lose that signal. If, let's say, you had another cytokine that was around here, this IL-6 cytokine was around this area, yeah, I would be hesitant because it's not in the linear range to say, I believe it's exactly 900 picograms per mil. But then again, if the standard curve would cut off the data here, it would just say above a certain level. And so you wouldn't get much out of that. You're much better off looking at the actual number in MFI than getting above 5,000 or above 900. Similarly here, if you would get below 1.23 picograms, that's not going to help you much. What do you think? I didn't get that. Abrogated beads? Aggregated. Oh, okay. So the aggregated beads are taken care of by the fact that we gate out the doublets. The aggregated beads are gated out, and I can show you in the demo. The spectral addresses are actually a range of spectral addresses, and those are the gates. You can think of the bead as a cell, right? And there's a gate that conforms to IL-2. Now, within a certain range, that is considered what you're going to be measuring. And the ones on either sides are going to be either aggregates of beads or doublets or just messy particles that are not going to be read.
But in some samples,
you get
specific aggregation of certain beads.
Yes.
That can be a problem. And there isn't.
There's no good solution to that.
I think even sometimes in the standard curves, and they give quite a lot of aggregated beads, but we still get a good standard curve.
Yes. The standard curve is usually better.
Some plasma and other kinds of tissue homogenous, sometimes they have something that aggregates certain beads and not all the beads.
If you're treating samples also with antibodies, but though from our experience, some people use different antibodies, and that sometimes causes aggregation and reduces the availability of some of the beads and aggregation, and then you have a problem which sometimes you can solve with dilution, but in the standard curve, we haven't seen that.